This presentation is going to give you a quick introduction to the microscope. Our next unit of study is cells, so it's important to make sure that we understand how we are even able to see cells and understand how we've been able to learn about what makes up a cell, the parts of a cell, and how cells work. And the major piece of technology that has come about or that has been refined to learn about cells is the microscope. So in this presentation you're learning about both the types of microscopes that are out there and the parts of the microscope. Microscopes are tools for what we can see. Take a look at this infographic. If you start back here, you're looking at the largest item that we can see, or a large item that we can see. Humans. Anything from the size of a human, and obviously bigger than a human, anything from the size of a human to down to the size of a hair we can see with our human eyes without needing any sort of tool to help us see them. If you look in this next range, we use light microscopes to help us see things from the size of insects all the way down to the size of something as small as a bacteria. Notice here, a cell is larger than a bacteria, and we can see both of these things with a simple light microscope. And then there's this third category of microscope, or second category of microscope, called electron microscopes. These allow us to see things that are really small. You can certainly see cells with electron microscopes, and you can see them in really, really fine detail. But then you can use electron microscopes to see things all the way as small, almost as small as an atom, but not quite that small, at least not yet. I'm sure there are scientists out there working on powerful enough microscopes to allow people to see single atoms. But right now, basically the smallest we go is being able to see a small molecule with a microscope. So we have four types of microscopes. A light microscope, this is typically what you would find in schools. A stereoscope, which is just another type of light microscope. A scanning electron microscope, and a transmission electron microscope. Let's start with the light microscope. Again, this is typically what you would find in schools. It uses more than one lens to magnify objects. The lenses of the light microscope are found up here in the eyepiece and down here in what are called the objectives. These lenses bend light to make the object beneath them appear closer. So what you do is you place a slide here on the stage, and you can see there's a hole here. The light shines up through that hole, goes up through the specimen, through the objective, through the tube, through the other lens in the eyepiece, and to your eye. And in that process, the way the lenses are shaped make the object appear larger. Common magnifications for a light microscope are 40 times, 100 times, and 400 times. That means at its smallest magnification, you are looking at your specimen 40 times bigger than it normally is. You can also add something called an oil immersion lens, which means you put a drop of oil on the slide, and that cuts out any airspace between the slide and the objective, and that improves the quality of focus and the magnification to some extent. So talking about magnification, your microscope has three magnifications: scanning, low power, and high power. Each objective will have its magnification written on it. In addition to this, the ocular lens, or the eyepiece, also has a magnification. So to find the total magnification, all you have to do is multiply. If the objective is 4 and the ocular is 10, the total magnification is 40. So this is where we get the 40, 100, and 400. Now let's talk about another kind of light microscope called the stereoscope. With this microscope, the light comes from this part right here. Notice that's above the object, not underneath it. This microscope allows for binocular viewing of larger specimens. You can see there are two eyepieces. It means you look through it with both eyes. It has less magnification than a normal light microscope. It only magnifies 10 to 20 times, but that's not that big of a deal because you're usually viewing specimens, objects that are much larger than what you would view on a light microscope. You can use thicker things, like you can look at an entire insect, not just a slice of an insect. And the major advantage to the stereoscope is that it creates a 3D view of the specimen. Now let's move on to the other type of microscope, the electron microscopes. The first one is the scanning electron microscope. When you scan something, you're just looking at the surface of it. So you can see some photographs of scanning electron microscope images of a variety of things, an insect, a pollen grain, and some other things. Scanning electron microscopes have much higher magnification than light microscopes. They don't use light waves, instead they use electrons, which are negatively charged electrical particles, to magnify objects up to 2 million times bigger than what they actually are. You can see the SEM, or the scanning electron microscope, creates a 3D image of the specimen, but unfortunately, because of the process that is required to put the object into the machine, you can't view anything that's living. The process that's required will kill the object. With a light microscope, either a stereoscope or a regular light microscope, you can view things that are actually alive and moving. Another type of electron microscope is a transmission electron microscope. This gives you an idea of the size of a normal electron microscope. They're really big. It's not something that you can carry around from room to room. This microscope also uses electrons, but instead of the electrons bouncing off the surface of the object, they actually pass through really thin slices of the specimen. And it allows you to view the inside parts of the organism. Here's an image from a transmission electron microscope of a cell. Notice that you see the inside of the cell and not the surface. The surface of the cell would be out around here. But all of these things, that's probably the nucleus. Here we've got some other parts to the cell. You've got a variety of things. Each of these is a part inside that particular cell. Okay, let's go over microscope parts. Use this diagram and this explanation to help you finish your assignment, but keep in mind the order that I go over the parts here does not match your assignment. Your assignment has a separate labeled microscope for you to write in the names of the parts. So let's start up here at the top with the eyepiece. This is where you look through the microscope. The eyepiece is connected to the body tube. That just provides the proper amount of space for the light to travel through. This piece here is the arm. It's what holds up the eyepiece and the body tube. Then you have a really important piece, the revolving nose piece. The revolving nose piece is called that because it revolves, it turns around, and it lets you have more than one objective lens in order to kind of increase your magnification without having to get a totally separate microscope. These three things attached here are the objectives. The shortest one would be the low power objective, the longest one would be called the high power objective, and the one in the middle would be called the middle power objective. Sometimes you'll also hear them called scanning, low, and high power. Now let's move on to these two knobs. When you put a slide here, you've got to be able to get the slide in focus. You start by focusing it with the coarse focus, or the coarse adjustment knob. The coarse focus actually raises the stage up and down. When you turn that knob, the stage actually moves up and brings the slide closer to the objective, and thereby makes it more focused. Once you have it generally in focus, then you can use this fine adjustment knob, or the fine focus, to really clarify what you're seeing, really bring it into a nice sharp focus. The stage is the platform that you put the slide on. Notice there's a hole in the stage right here. That's where the light shines through. And the stage clips are used to hold the slide in place so it doesn't go flying off if you happen to bump your microscope, because then it becomes really difficult to find what you were looking for. The base is at the bottom of the microscope. That's where all the electrical parts are stored, and kind of provides the support for it to sit on. Underneath the stage you have what's called the diaphragm. The diaphragm kind of looks like a camera shutter, and it opens or closes. It makes a bigger hole or a smaller hole, which adjusts the amount of light that goes through the stage. Sometimes with more light it's easier to tell what your specimen looks like. Sometimes it's easier to tell what your specimen looks like with less light. And finally here you see the light source. This is an electric microscope. Some microscopes use mirrors to reflect light up through the eyepiece, but this one has an electric switch where you turn the light on or off with a light bulb. Okay, this brings us to the end of the introduction to microscopes. If you have any questions, please make sure you send me a message.